So if we look at the electron transport chain, which are the proteins that are embedded in the inner mitochondrial membranes, in the cristae of the inner mitochondrial membranes. This is a complex process. We're going to kind of work through it in a couple of steps. So we have three main protein complexes. You'll go in a little bit more depth in this if we talk about it in cell biology. We've got these NADH and FADH that have been formed from the TCA cycle and from glycolysis, right? So all of these reduced electron carriers are coming in to the mitochondria or being produced in the mitochondria as end products of either glycolysis or the TCA cycle. And they get next to these electron transport chain proteins, and the electron from the reduced form, let's look at just NADH first, is handed off to the electron carrier in this protein complex, at which point NADH has lost its electron and has been oxidized. So NADH is oxidized in that it loses its electron. Remember, lose electron, oxidized. As that process occurs, a hydrogen ion from the inner mitochondrial membrane space, from the matrix, is moved across that pore, through the pore of the ETC protein, complex 1, into the inner mitochondrial membrane space. Those electrons, so for every NADH that interacts with this complex, the NADH gives up its electron, and a hydrogen ion is pumped through from the matrix into the inner mitochondrial membrane space. That electron then gradually loses energy as it proceeds through the electron transport chain complexes. And each time it moves through one of these complexes, it's losing a little bit more energy, but as it moves through, another hydrogen ion moves out. So it moves through this one, we get a second hydrogen ion, so we get one here, we get one here. It moves through this other protein complex, 4, and we get a third hydrogen ion moving through. So the electron, so for every NADH that is oxidized, 3 protons are pumped into the inner mitochondrial membrane space. 3 protons, 2 IM space.